Hello friends, today we are going to discuss about stellar evolution in vascular plants. Before we go to the stellar evolution, first of all we should know about the stele and what are the components of stele. Basically the components are xylem, phloem, pericycle. Sometimes it is medullary ridge and pith. If you see when we see under microscope a TS of dicot stem, it usually looks like this. And whenever we see in the secondary growth of a dicot stem, it looks like this. So however, this is the central region which is called as stele. Let's go to the stellar theory. The stellar theory is first given by the Van Tegham and Doliet in 1886 in their stellar theory. As the components they described the xylems and phloem. Tissues like pericycle, sometimes medullary ridge, pith and pith are also component of the stele. Cortex and stele are the two fundamental parts of the shoot, and these are separated by the endodermis. In this picture you can see you can differentiate the cortex and primary xylem, pith, all these things are described here. Leaf traces in higher vascular plants like pteridophytes, gymnosperm and angiosperms play an important role in vascular system. Leaf traces. So here it is mentioned in the leaf trace and leaf cap. So how do these leaf trace and leaf cap we can find from a TS? If you see this picture, this is the central cylinder of stele and these are the leaf traces. And when you take a TS of this region, then obviously we will get a leaf cap here, and these leaf caps and leaf traces result to distinguish different type of stele. Stele may be of different types in higher plants. These are basically Protostele, Siphonostele and Solenostele. So as we know, the Protostele is the most primitive. From the name itself you can understand that is Protostele, that is primitive type, and perhaps the Siphonostele is derived from Protostele, or it may be a modified version of Protostele, and Solenostele is the advanced form of Siphonostele. However, different authors they have classified this Solenostele under Siphonostele. Protostele, if you see, it is the simplest stellar organization and it is a primitive type as I told, and it is the solid core of xylem at the center surrounded by the phloem, pericycle and endodermis, and pith is present in this type of stele. Majority of pteridophytes show protostele condition in the rhizome, stem and roots. If you classify the protostele, it may be of 5 types, that is haplostele, actinostele, plectostele, mixed protostele and mixed. So now describe about the haplostele. So how it is looks like and what are the basic characters. So haplostele, if you see the examples are Selaginella, Glycenia and Lygodium, and usually this kind of stele is usually found in the fossil genera like Hania and Horneophyton. And protostele, it is a smooth core of xylem surrounded by phloem, and it is uniform layer actually. Then if you see the actinostele, it will be usually the star shaped, or the central xylem core will be star like or stellate like, and the phloem is not continuous here. The phloem is not continuous here because it is interrupted by this asterisk arms, and the examples are asterxylon, xylotom, lycopodium, ceratum, etc. And this is a figure of lycopodium ceratum, and these are the diagrammatic feature of haplostele and actinostele. Then come to the plectostele. Plectostele, it means that the xylems are plate like arranged inside phloem, and it is given by Zimmerman in 1930, and the example that is Lycopodium clavatum, and this is the transfer section of Lycopodium clavatum. And here it is mentioned that this is the leaf trace, and these are the phloem, metaxylem, protoxylem, pericycle, endodermis. This is cortical region and this is epidermis. And this is the natural photograph of Lycopodium clavatum, and this is a diagrammatic features where these xylems were shown in plate-like structure, and these are the phloems. Likewise, mixed protostele is the xylem occur in several groups inside phloem, and the examples are Lycopodium cernum. Likewise, there is no such differentiation between mixed protostele and mixed protostele with pith. The only difference is patches of parenchymatous region occur in association with xylem. That means in association with xylem there will be patches of parenchymatous tissue which represents the pith. The examples of Hymenophyllum demicium, Lepidodendron, Serraginoids. Then come to the Siphonostele. Siphonostele, it is a stele with pith at the center, and the central core of pith is surrounded by xylem. Advanced type, it is advanced type over the protostele. If you see the origin of Siphonostele, it is believed that it is derived from the protostele by the formation of pith, and it is the central part of xylem in the protostele is gradually replaced by parenchyma cell. So there are two beliefs on the gradual replacement of parenchyma. Those are interstellar origin of pith and extrastellar origin of pith. So considering the origin of pith in interstellar form, it is stated that the pith region in Siphonostele condition is derived from the core region of the protostele. That means the core, some cells of the core region, they may be converted to parenchymatous cell and they form the pith region. However, the extrastellar region of pith, saying that the ground tissue parenchymatous cells may invade inside to the core region. That is, the invasion of cortical parenchymatous cells into the stele, which led to the pith region in Siphonostele conditions. If we classify the Siphonostele, it may be two types, that is ectoploic Siphonostele and amphiploic Siphonostele. Ectoploic, the word itself describing, that is, the phloem is outside of the xylem. That means phloic, that denotes the phloem, and ecto means the outside. Collectively we can say the phloem present only on the external side of the xylem, and phloem is surrounded by pericycle and endodermis, and the examples are Osmunda and Syzygia. In this picture you can see the phloem region, this is the phloem region, is just outside the xylem region. That's why it is ectoploic. Similarly, the amphiploic means, if you split the word, amphi means two and ploic indicating the phloem, that is phloem on both sides of the xylem. From this picture you can see this is the xylem and the phloem are on both sides. That's why it is amphiploic. This is also the diagrammatic picture of ectoploic Siphonostele, and this is amphiploic Siphonostele, where the xylem is surrounded by phloem, and here the xylem is surrounded by phloem and inner phloem and outer phloem. Then come to the Solenostele. If you see Solenostele, that is, it is differentiated by the presence of the leaf gap. Just nothing but the Siphonostele having leaf gap. This is Siphonostele and this is Solenostele. In Siphonostele there is no leaf gap, but here it is a leaf gap. Got it? Then I have already described that how the leaf gap is produced due to the presence of this leaf stresses, and the leaf gap is here. So further, if we classify Solenostele, it may be of ectofloic Solenostele, amphifloic Solenostele, dictyostele, polycyclic stele, Eustele, or atactostele. So let's have a discussion one by one. So if you see that the ectofloic Solenostele and amphifloic Solenostele, these are nothing but a just slight modification of ectofloic and amphifloic Siphonostele. Here these pictures, if you see, these are ectofloic and amphifloic Siphonostele, and these are ectofloic and amphifloic Solenostele. The only difference is the leaf gap present in ectofloic and amphifloic Solenostele. All the other things are as such as in ectofloic and amphifloic Siphonostele. The examples you can cite, that is you can remember, that is adiantum, pedatum. Then come to the dictyostele. Dictyostele, it is a solenostele that is broken into separate vascular strands due to the leaf gap. So these points are the leaf gap which is separating the vascular strands into different meristele. So each strands are called as meristele, and it is produced because of the leaf gap. Then come to the polycyclic stele. If you see the polycyclic, the word itself describing, that is poly means many, cyclic means ring, that is many rings. Here you can see many rings of vascular strands and with a leaf gap, that's why it is polycyclic Solenostele. Another one is polycyclic dictyostele. That means it may be the dictyostele with many rings. That means different strands or separate meristeles due to leaf gap, and this vascular strand is of multiple rings, as you can see here in polycyclic dictyostele. Here this is a ring of meristele, this is a ring of meristele. So these meristele were produced because of the leaf gap. So these are the leaf gaps here, 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 and likewise these are also the leaf gaps. So these leaf gaps produces different meristele in case of polycyclic dictyostele. So this is all about dictyostele and polycyclic stele. Now come to the Eustele and atactostele. Eustele also, if you see the word, that is Eu means true and stele means the central core. In this picture, a series of vascular strand is dissected by different leaf gaps, and due to this leaf gap, this vascular strand is broken and collateral vascular strand exists. This type of stele you can find in dicot stem primary structure, and this is the xylem part, this is the phloem part. Likewise, in monocot stem, the vascular bundles are in scattered form. It gives more mechanical support to the monocot stem, and this is most advanced form in Solenostele. And this is the summary of all slides, that means different type of stele, that is Protostele, Siphonostele and Solenostele. The basic difference between three is in Siphonostele with no leaf gap. In Solenostele there is with and leaf gap. Further, if you classify Protostele, there may be haplostele, actinostele, plectostele, mixed protostele and mixed protostele with pith. Likewise, in Siphonostele, that is ectofloic Siphonostele, amphifloic Siphonostele, and we have described that is in Solenostele, that is ectofloic Solenostele, amphifloic Solenostele, dictyostele, polycyclic stele, Eustele and atactostele. This is the haplostele or protostele, and all these apex parts may be derived from this protostele in a sequential manner. So if you see, the protostele may give rise to the ectofloic siphonostele and siphonostelic eustele conditions may be evolved from the ectofloic siphonostele, and eustele may be evolved from siphono-eustelic condition. Likewise, amphiploic, the ectostele may be evolved from dictyostele, dictyostele may be from amphiploic siphonostele, and amphiploic from protostelic conditions. Likewise, actinostele, plectostele, these are evolved from the haplostele condition. So these are 5 questions for you guys. So try to answer all these 5 questions. Thank you very much guys for your patience hearing. Hope this lecture will give you some information. Like, subscribe and share this article, which will inspire me to create some more videos from your syllabus. Thank you very much.